Welcome dear students. In this video we are going to learn about TGF beta signaling pathway. So at first you have to remember the TGF beta family member. Because TGF beta family members are actually acts as a ligand. Example of this TGF beta family are TGF beta 1, TGF beta 2, TGF beta 3. Others like activin, inhibin, nodal, VG1, bone morphogenetic protein BMP. There are different type of BMP are there, BMP2, BMP3, BMP4, not BMP1 because BMP1 is an enzyme called actually a protease. So BMP in short, bone morphogenetic protein. Then AMH, that means anti-Mullerian hormone. Another is called GDNF, that means glial derived neurotropic factor. So these are the TGF beta family members. So these actually acts as a ligand of this TGF beta signaling pathway. So these are the ligand. Okay, any of them can activate the TGF beta signaling pathway. Specifically I want to mention some facts that TGF beta actually, these ligands actually bind as a dimer. So TGF beta actually bind as a TGF beta binds as a homodimer or heterodimer. Specifically I'm mentioning here TGF beta 1, 2, 3. The TGF beta ligands can be TGF beta 1, 1 or TGF beta 1, 2 or so on. Okay, so it could be homodimer or heterodimer. Another important fact about TGF beta signaling pathway is TGF beta receptors are like called R1, R2, R3. These are three TGF beta receptor. These receptors are actually acts as a serine threonine kinase. So these are actually some enzymes. They have cytosolic domains, I will show you, that actually acts as a serine threonine kinase, specifically R2 and R1. Okay. Another fact about this TGF beta signaling pathway is that TGF beta actually activate a specific transcription factor called Smad, S-M-A-D, Smad. This word actually comes from two different molecules from different organisms. Like the SMA, Smad, is come from the C. elegans, means Sinorhabditis elegans. And the other word that is, so SMA come from C. elegans and MAD, mat, from Drosophila. So these two word actually, this SMAD is the amalgamation of these two word, SMAD. Now this SMAD is a transcription factor and this SMAD can be of three types. One is called R-SMAD, that means they can, this SMAD is actually activated by the receptor SMAD. Example like SMAD 2, SMAD 3. There is some co-SMAD or C-SMAD that is called co-SMAD, that is called SMAD 4, and another is called I-SMAD. These are the inhibitory SMAD like SMAD 6 and SMAD 7. Okay, TGF beta pathway basically activate some genes. Example, they activate this pathway, actually activate gene. Example are like plasminogen activator inhibitor 1, called PAI1. This protein, this plasminogen activator inhibitor is actually a molecule that actually inhibit serine protease. What is the significance of PAI1? I will talk about in later. So at first you know the basic facts about the TGF beta signaling pathway. Like this TGF beta pathway, like this Smad, actually activate these genes. Another example of gene called P15INK4B. This protein also inhibit cell cycle at G1 phase because it inhibits some CDK molecules, G1 phase. And the third protein I can mention here that actually, or a function of this TGF beta signaling pathway is for stem cell, so that the stem cell remains undifferentiated in conditions. So TGF beta also function in stem cell undifferentiation. So these are all some basic facts about the TGF beta. Now I will show you how TGF beta signaling pathway actually occurs. What happens? Suppose this is the plasma membrane of a cell. This is the plasma membrane of a cell. This is the bilayer. This is a cytosolic lesion, cytosol. This is a plasma membrane. There are two ways the TGF beta pathway actually can activate. The first is there is a, let's suppose this is a transmembrane protein. This is called R3. This protein is actually a proteoglycan. This protein, the R3, is also known as beta glycan because this is a proteoglycan. Proteoglycan means a protein associated with sugar molecules, specifically called glycosaminoglycans, GAG, GAG protein, like heparan sulfate or chondroitin sulfate. These are the GAGs, glycosaminoglycans. When these molecules associate with proteins, we call glycoprotein. So R3 is actually beta glycan. R3 means beta glycan. This is actually a receptor for the TGF beta I have mentioned here, but it has no kinase activity. What happens? The two TGF beta homodimer or heterodimer. Let's suppose this is a TGF beta homodimer or heterodimer, whatever. It actually binds with this R3 receptor. Then this R3 actually represent or concentrate this TGF beta so that R2 is another receptor, and I am showing this structure as a cytosolic domain of the receptor. Here the difference is, in case of R3 this is a single transmembrane glycoprotein, but R2 is a dimer. Now this R3 actually represent or actually give this TGF beta to the R2, so it can donate the TGF beta to R2, or in some cells TGF beta dimer bind with this R2 directly. So there is two way, one way and another is directly it bind with R2. But one thing you have to understand, this is the informations you can remember. This R2 is actually a constitutively activated protein, constitutively show kinase activity. That means this R2 proteins, whether it binds with TGF beta or not, it is actually active in state. So it is always active. So this kinase activity is always active. So it is called constitutively activated, whether it is bind with TGF beta or not. But then why this protein cannot activate this pathway all the time? Yes, there is a questions. The answer depends on the binding of TGF beta with the R2 receptor. When TGF beta dimer bind with this R2 by this way or this way, by donating from the R3 or directly bind with the R2, there is a change in conformations that actually helps in the docking of this whole molecules with other, let's suppose this is also the kinase domain receptor called R1. You have to understand this. I have mentioned that R2 is a constitutive kinase activity, has constitutive activity. That means whether it binds with TGF beta or not, it is active. Then why cells always do not perform TGF beta signaling? The answer is when TGF beta, this is TGF beta, when TGF beta bind with this R2, it changes its structure so that it acts as a docking site for the R1. Then R1 and R2 actually come close to each other, very close to each other. They do not remain very distantly. They actually associate. They associate with each other. So they come very close. Let's suppose this is the R1. And what happens? This R2 cytosolic domain, this is the cytosolic domain of the R2, has kinase activity that actually phosphorylates particular portion of the R1 receptor so that it gets activated. So by this way, this kinase activity actually activates this protein kinase activity. So R1 is actually inactive in condition, then it becomes activated. So the kinase activity of R1 gets active when this R2 phosphorylate particular serine or threonine residue of the R1. Then R1 is capable to phosphorylate other proteins at the serine or threonine kinase, serine or threonine positions. You have to remember, because this is the serine threonine kinase enzyme. This has the property. Now the second part of this signaling process is Smad protein. I have mentioned that TGF beta family activates the transcription factor called Smad. Here the R Smad called the Smad1 and Smad2, whatever may be, it can be Smad2 or Smad3. Smad2 or Smad3 have a very delicate way to remain inactive. That is, let's suppose this is Smad2 or Smad3, whatever it may be. They look like this. They have two domains called MH1 and MH2, and they actually linked by a linker domain. I have shown in this way. The main fact about this thing is when this Smad2 or 3 is inactive in conformation, that means when there is no TGF beta, then they remain as this. MH1 and MH2 come close to each other, remains close to each other. And the MH2 actually block a region or domain of this MH1 that is called NLS. That means NLS means nuclear localization signal. In my protein sorting video, I have mentioned what is the significance of NLS. NLS actually helps in the import of proteins into the nucleus. So it is called nuclear localization signals. There is a particular protein called importin. Importin protein can bind with this NLS and bring the protein to the nucleus. When this system is off, when there is no TGF beta, this Smad is inactive in conformations, then MH2 binds with this MH1 and there is a NLS domain in the MH1, so it is actually masked. Now in this MH1 domain there is also a region called DNA binding domain. This is also inactive in conformation, it cannot bind. Another thing is this Smad2 or Smad3 can bind with Smad4, that is co-Smad. I have mentioned as a C-Smad, co-Smad, Smad4. What happens when this thing occurs, the TGF beta binds, then R1 gets activated. This R1 then phosphorylate, a two particular serine residue in the MH2 domain. When MH2 domain gets phosphorylated, it changes its conformations, and how it looks when it gets phosphorylated, it looks like this. MH2 that is phosphorylated, this is the linker, okay, let's suppose this is the linker, and this is the MH1 domain. Now you can see there is no longer, it binds with the MH1, so the NLS domain is now open, so that importin protein can bind with this MH2 protein, this is importin, and can take this protein to the inside the nucleus, because this is actually a transcription factor. Now another thing is that this SMAD2 or SMAD3, whatever it may be, is bind with another SMAD2 or 3 and with the co-SMAD. So in a complex there should be 2 SMAD2 or SMAD3 and 1 co-SMAD, that means SMAD4. So always the complex should be 2 SMAD2 or 2 SMAD3 and 1 SMAD4. Here, when these serine residues are phosphorylated, another SMAD2 or SMAD3 bind with this domain. This could be the MH2 domain, and there is free MH1 domain. Then this MH2 domain is actually phosphorylated. Now this domain is actually attached with the MH2 of the, and this is the MH1 by the SMAD4. You have to understand this. So there is the 3 molecules, 2 SMAD or 2 SMAD3, SMAD2 or SMAD3 and 1 SMAD4. It could be the SMAD2 or SMAD3 and other would be the co-SMAD called SMAD4. Now these 3 protein actually enter into the nucleus. They enter into the nucleus and there is a gene. They bind with this gene as a complex. Let's suppose this is the complex, and activate the gene. Example I have mentioned, PAI, plasminogen activator inhibitor 1. This is actually a serine protease inhibitor. When this protein actually gets transcribed and translated, this protein inhibits the serine protease, and that serine protease actually digests the extracellular matrix. So when this PAI protein actually gets produced, then the serine protease cannot perform its function. So extracellular matrix remain intact. And what is the significance of extracellular matrix? So the first thing is TGF beta, when it is secreted, it actually remains inactive and bind with the extracellular matrix proteins. When serine protease are secreted, the serine protease digests the extracellular matrix so that the TGF beta can actually be released. Actually, the structure of TGF beta is like this. It has a N-terminal prodomain and the active domain. This N-terminal prodomain actually associates with some extracellular matrix protein so that the TGF beta, real TGF beta, cannot move. So when serine protease digests this extracellular matrix, then this protein TGF beta can freely move from the extracellular matrix to the target cell. So when serine protease gets inhibited, then TGF beta cannot be released. So when a TGF beta signaling starts, it actually inhibits its own signaling pathway. Another is called P15 INK, I have mentioned 4B, that is actually inhibitor of G1 pathway. Another, it can associate this TGF beta SMAD protein, actually SMAD2 or 3 protein with the SMAD4, can associate with OCT4, SOX2 and NANOC. This is actually helps in the undifferentiated state of stem cell. So this is the pathway. Now I will show you how this pathway is actually negatively regulated, because there should be some negative regulations. This is called negative feedback loop. So in this pathway also there is a negative feedback loop. The negative feedback loop is performed by 2 proteins. One is called SNON, another protein is called SKI. SKI, that means Sloan Kettering Institute, Cancer Institute. Okay, this is actually a very famous cancer research institute in the New York. So SKI and SON can inhibit this pathway. And another is called just iSMAD. iSMAD, inhibitory SMAD like SMAD6 or SMAD7 can inhibit the TGF beta pathway. I will show you how this occurs. Especially the SKI1 is actually known how it actually works. SKI1, so let's suppose this is a gene. So when the TGF beta pathway is activated, then SMAD, let's suppose this is a SMAD protein, SMAD 3 or 2 and with the SMAD 4, these 3 complex actually bind with the DNA. Then after a few time, then another protein called SKI, SKI means Sloan Kettering Cancer Institute. From that name it actually comes because it acts as a proto-oncogene. SKI, okay. So this SKI protein binds with this SMAD, so this is SKI. SKI associate with another protein called NCOR protein. NCOR protein associate with another protein. These are the building block. It's called MSyn3A. You don't have to remember this name. Just remember the SKI. But the thing is that it associates with NCOR. NCOR associates with MSyn3A. Then ultimately MSyn3A associate with a very important protein that actually helps in the inhibition of this pathway, that is called HDAC, histone deacetylase. This histone deacetylase, what can it perform? It actually deacetylates the nucleosome. When nucleosomes are deacetylated, the nucleosome actually tightly packed and form. So HDAC helps in the heterochromatizations, heterochromatin formations, because we know in case of heterochromatin the DNA is actually locked in state, so it cannot be transcribed. So this is one example of a negative feedback loop of this system. Another is iSMAD. In the book of Bruce Albert, it has been said that this SMAD6 and SMAD7 can perform its functions, inhibitory functions, by 3 ways. One is it compete with rSMAD, SMAD2 or SMAD3. Another way, it recruits, this 2 protein recruits a ubiquitin ligase. Ubiquitin ligase is an enzyme that actually helps in the ubiquitinations of a protein. The ubiquitin ligase name is SMURF. The third way to inhibit this pathway, that this 2 protein can activate a protein phosphatase, and ultimately this protein phosphatase actually dephosphorylates the receptor of phosphate group. Because I have mentioned that when the R1 receptor of the TGF beta family gets phosphorylated, then it becomes active, then it can phosphorylate the SMAD protein, SMAD or rSMAD, SMAD2 or SMAD3. Now when this gets dephosphorylated, because it's a phosphatase, when protein phosphatase gets activated, it dephosphorylates the R1 phosphate group, then it cannot no longer phosphorylate other SMAD proteins like SMAD2 or SMAD3. So this is the negative feedback loop of TGF beta family. Hope this video will help you. If you have any doubt about this topic, you can write me in the comment section. Thank you for watching and always keep learning.